Very good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET World. So yes, today we are in our last session of Sexual Reproduction of Flowering Plants chapter, which is a major chapter both for EAPCET and for NEET. So today what we are going to learn is post-fertilization events, ma'am. After post-fertilization events, we will be exercising the EAPCET previous year questions. So let's get started with post-fertilization events. What are the major events that happen after fertilization in an angiosperm? Okay, that is the concept. So what happens is, the very first thing I told you, that inside the embryo sac, after the fusion of one male gamete with an egg cell which forms a zygote which is diploid, and one more male gamete with an polar nuclei, this forms out to be primary endospermic nucleus, which is triploid. Okay, this is triploid. So after fusion, these are what zygote and primary endospermic nucleus will be formed, where zygote will be developed into an embryo, which comes under embryo development. Primary endospermic nucleus will undergo sequential nuclear divisions and cellular divisions, which will form out to be what? Endosperm. So this comes under what? Endospermic development. So endosperm development will prior to the embryo development here. First endosperm development will happen, and then embryo development will start off, because the endosperm which is developed will be consumed by the embryo to be developed. Okay, embryo will the embryo development, this process will be consuming what? Endosperm, okay, which is there. So here you see, after which seed formation and fruit formation, these two will be taking place simultaneously. Seed formation and fruit formation will take place simultaneously. Okay, seed formation and fruit formation will take place simultaneously, whereas endosperm development will be prior to the development of embryo. And inside the embryo sac, so first after fusion, immediately what happens? The antipodals and the synergids will be degenerated. Okay, antipodals and synergids will be degenerated. Fine, right. So this is about post-fertilization events. And then you see endosperm, which is triploid. So endosperm is not same in all the plants, beta. In few of the families, endosperm is absent. In few of the families, endosperm is diploid. In few of the families, endosperm is triploid, but then the composition can be starchy endosperm, cellulosic, proteinaceous, or oily endosperm. So based on that, we have different types which are very important for our objective. So see, endosperm which is triploid. So here these are absent in, endosperm is absent in few families like Orchidaceae, Podostomaceae and Trapaceae. In these families, endosperm is absent. Okay, endosperm is absent in Orchidaceae, Podostomaceae and Trapaceae. Then stony endosperm you can see in betel nut, that is areca nut, and in date palm, that is Phoenix dactylifera. Okay, so stony endosperm is observed in what? Betel nut, that is areca nut, and in date palm, that is Phoenix dactylifera. Diploid endosperm is observed in Oenothera. Okay, diploid endosperm is observed in what, ma? Oenothera. So here you see first cereals. For example, in rice and wheat you can see starchy endosperm. Okay, cereals like rice and wheat will be having what? Starchy endosperm. Cereals like maize has or possess proteinaceous endosperm which has aileron layer. Now when we are learning or when we are looking into the structure of monocot seed, you will get a clear idea about starchy endosperm layer and aileron layer of endosperm. Next, castor and coconut will be having what? Oily endosperm, from which we will get the coconut oil or castor oil. And then we have ivory palm which has cellulosic endosperm, date palm will be having hemicellulosic endosperm. Okay, these are the compositions of the endosperm beta, you have to remember. And endosperm is absent in the families like Orchidaceae, Podostemacea and Trapeaceae. Stony endosperm is present in few nuts like betel nuts, that is erica nut, and date palm, that is Phoenix dactylifera. Diploid endosperm is observed in what ma? Oenothera. Clear till here? Any doubt anywhere? Yeah. Now we will see endosperm development. Endosperm development is of two types. One is nuclear endospermic development. Second is what? Cellular endospermic development. See here. Okay. So the first one is what ma? Yeah. Here we go. First one is nuclear endospermic development. Second is cellular endospermic development. So here first, that is triploid primary endospermic nucleus, after this formation sequential mitotic divisions will happen, but then only karyokinesis takes place. Okay, only karyokinesis occurs, only karyokinesis. Fine, right? So after continuous karyokinesis, later cell wall formation will be formed like this. Later, at last, cellularization will take place. So this type is known as nuclear endospermic development, where primary endospermic nucleus will continuously undergo nuclear divisions, and at the end cellularization will take place. And next you see here ma, cellular endospermic development. Here what happens? A single nucleus or a single cell will start undergoing the sequential mitotic divisions, where at each cell division karyokinesis, cytokinesis both occur, that is karyokinesis will occur and cell wall formation also takes place parallelly. Okay, cell wall formation also occurs. Fine, right? So cellular endospermic development, wherein each cell will undergo karyokinesis and cytokinesis, where cell wall formation will take place after each cell division. This leads to the formation of cellular endosperm. The best example I can give you is coconut. Okay, coconut, where in coconut water that we drink, right, that is the result of nuclear endospermic development, where continuous karyokinesis will take place without cell wall formation. And the white kernel that we eat, okay, white kernel of coconut that we eat, that is the result of what? Cellular endospermic development. This is cellular endosperm and this is nuclear endosperm. Clear till here? Clear endospermic development and types of endosperms that you see. Here we go with fate of endosperm. In few of the seeds, endosperm is still persistent in the seed. You can observe in most of the monocots. Okay, most of the monocots, or monocot seeds will have persistent seeds, wherein endosperm is still persistent in mature seed. In a mature seed, endosperm is still persistent, and this is used up later after seed germination. Okay, best examples that I can give you about persistent endosperm is our endospermic seeds. So those seeds which still have endosperms, you can see it in most of the monocots. Only dicot example that I can give you is castor. Okay, most of the monocots will have what? Endospermic seeds only. So endospermic seeds are also addressed as albuminous seeds. Albuminous seeds or endospermic seeds. albuminous seeds or endospermic seeds. If you see non-endospermic seeds or X-albuminous seeds, where complete endosperm is consumed by the seeds. Okay. So, endosperm may be either completely consumed by the embryo during the embryo development before seed maturation, and food reserves get stored in the cotyledons of the seed, cotyledons which are addressed as embryonic leaves. Okay. In the embryonic leaves, that is cotyledons, food will be stored. So, here you see these are what X-albuminous or non-endospermic seeds. Okay. These are X-albuminous or non-endospermic seeds. Fine, right? Okay. Examples here include pea, bean and groundnut. So, here if you see in the monocot seed, most of the monocot seeds are albuminous seeds or endospermic seeds, wherein endosperm is not completely consumed by the embryo development. Whereas, if you see, so here we address this as what? Persistent endosperm. If endosperm is fully consumed or mostly consumed during the time of embryo development, and in the seed you do not find anywhere, endosperm you do not find anywhere, such seeds are addressed as what? X-albuminous or non-endospermic seeds. Examples here you can take is pea, bean, groundnut and or most of the dicots. Okay. Here you can consider this as what? Most of dicots. Clear till here? Any doubt anywhere Yeah. Next you see? Yeah. Here we go with embryo development. Okay. So, first product of fertilization is what beta? First product of fertilization is zygote. Zygote will undergo sequential mitotic divisions and it will form embryo. So, here we go with embryo development both in dicots and monocots. First we will see it with dicots. Okay. Dicot embryo development. First you see diploid zygote. Diploid zygote will first undergo a transverse division. A terminal cell is formed at the chaliceal end and a basal cell is formed at the micropylar end. Clear, right? Terminal cell is formed at the chaliceal end and a basal cell is formed at the micropylar end when the zygote will undergo first transverse division, after which, okay, this terminal cell, this basal cell which is long will undergo sequential transverse divisions wherein 6 to 10 cells are formed. This is addressed as what? Suspensor cell. Okay. This cell, what do you see? You see this basal cell, just a second. So, zygote here will first undergo transverse division and it will form a terminal cell at chaliceal end, basal cell at micropylar end. Clear till here? And here we go with first, this basal cell will undergo sequential divisions forming many celled condition. This is suspensor. Okay. So, the apical region cell is addressed as haustorial cell or vesicular cell. Vesicular cell, haustorial cell or vesicular cell. The terminal cell of suspensor is haustorial cell or vesicular cells, and the last cell of the suspensor is known as what? Hypophysis. Clear, right? Terminal cell will undergo parallel anticlinal divisions and periclinal divisions. What is anticlinal division? This perpendicular division is called as anticlinal division. Transverse divisions, parallel divisions are addressed as periclinal divisions. Both anticlinal and periclinal divisions will result in group of cells. Lower base is called as epibasal cells, upper one is called as hypobasal cells. Epibasal cells will result in the formation of cotyledons. Epibasal cells will result in the formation of cotyledons. Fine, right? Hypobasal cells will result in the formation of what? Hypocotyle. Hypocotyle. Fine till here? Any doubt anywhere? And this will later mature to globular embryo. Globular embryo will mature to what? Heart shaped embryo which will give rise to what? Complete dicot seed. Okay, here there is one doubt, ma'am, what are epicotyle and hypocotyle? Listen carefully, beta, I will explain you. First you see here epicotyle and hypocotyle is there, right? Epicotyle and hypocotyle are those cells, you can say initial cells, which will give rise to plumule and radicle. Epicotyle, these cells will give rise to plumule, plumule later matures to what? Shoot, okay, mature shoot. And hypocotyle, which these are the initial cells which will give rise to what? Radicle, and that will later mature to what? A complete root. Fine, right? Understood? So, epicotyle are the initial cells which comes out of the embryo. Epicotyle is nothing but initial cells that comes out of the embryo. Which will turn to the formation of what? Epicotyle will turn to what? First, that is plumule. This will later mature to what? Shoot. Same the case with hypocotyle, beta. Hypocotyle, radicle and root. Fine, right? Once again you look into dicot embryo development, wherein the zygote will first undergo a transverse division or a horizontal division, which will form the terminal cell and a basal cell. Here this is at the micropylar end and this is at the chalazal end. Here this cell will undergo 6 to 7 transverse division, which are addressed as the suspensor cells. Suspensor typical or apical cell is known as what? Vesicular cell or haustorial cell, and the basal cell of suspensor is known as hypophysis. Fine, right? Here the terminal cell will undergo sequential anticlinal divisions and periclinal divisions, forming what? Epibasal cells and hypobasal cells. This will later mature to what? A globular embryo. Globular embryo will mature to what? Heart-shaped embryo. This is what, ma? Globular embryo. And this is what, beta? Heart-shaped embryo. Fine, right? So, here you can see two cotyledons, which is dicotyledon. Cotyledons are known as what? Embryonic leaves. And here epicotyle will arise, this will later mature to plumule, plumule matures to shoot. And here you can see hypocotyle, which mature to radicle, radicle has at the tip what? Root cap, and this matures to what? Root. Dicot embryo development, is everything clear, ma? Any doubt anywhere? So, here the one who has doubt, is the concept clear, beta? Are you here in live? Next, monocot embryo development, which is very easy and simple when compared to dicot embryo development. Here we go. Monocot embryo development. Here also, first zygote will undergo a transverse division forming terminal cell and a basal cell. Basal cell will again undergo a transverse division forming out a suspensor cell at the micropylar region, terminal cell chalazar region, and a middle cell at in between both of them. This will undergo transverse division and vertical divisions forming out what? A globular embryo, okay, a globular embryo directly. So, here there are no two cotyledons, only a single cotyledon. Here you can see after globular embryo there is a single cotyledon, radicle and a suspensor cell, that is it. So, this is the simplest division that is followed by what? Monocot embryo development, simple division that you see. There is no heart shaped embryo that you can see in monocot embryo development, only globular embryo. Globular embryo. Fine, right? Now you see here monocot seed. Monocot seed, if you take a transfer section of this, here you can see a scutellum. Scutellum is known as papery cotyledon. Scutellum is known as papery cotyledon. This scutellum will drive out nutrients. This will drive out nutrients from endosperm which is present in the monocot seed. So, this endosperm can be proteinaceous endosperm, that is aileron layer, and starchy endosperm. From this, the nutrients are driven to the embryonal axis here. At the tip of embryonal axis, plumule is present. Covering the plumule, there is a protective sheet called as what? Coleoptile. At the basal region of the embryonal axis, radicle is present. Covering the radicle, the protective sheet which is there, that is coleorhiza. Coleoptile and coleorhiza are the protective sheets above the plumule and radicle that you observe only in the monocot seed, not in the dicot seed at all. Clear? Clear till here? Any doubt anywhere Vita about seed description? So, here you have a papery cotyledon, that is scutellum, which is driving out the nutrients from endosperm to the embryonal axis which is over here. At the tip of the embryonal axis, you have shoot tip which is plumule. Covering the plumule, you have what? Coleoptile, and at the base you have what? Radicle. Covering the radicle, you have a protective sheath, that is coleorhiza. Till here everything is clear, no? Any doubt anywhere, ma? Yeah. Next you see seed. Okay. So, seed here we have two types of seed. First you have to look into what are the parts of a seed. Seed consists of seed coat and cotyledons. Seed coat and embryonal axis you can say. Embryonal axis. Fine. And then cotyledons. Here a dicot seed will have outer seed coat that is known as testa and inner seed coat known as tegmen. Okay. Outer seed coat and inner seed coat. Outer seed coat of a dicot seed is testa and inner seed coat is addressed as tegmen. Fine, right? Outer seed coat and inner seed coat of a dicot seed. And then later monocot seed here. Monocot seed, if you see ma, you can see endosperm. So, here you have endosperm, embryo and cotyledons. Just a second. Seed coat, endosperm, embryo. This consists of cotyledons and embryonal axis. Clear, right? Okay. So, here if you see monocot seed, you have endosperm here, that is starchy endosperm covering the monocot seed. I am taking corn as an example, maize seed kernel. So, here if you see the outer endosperm is called as aileron layer which is proteinaceous and inner starchy endosperm is present. Here a papery scutellum which is driving out the nutrients from the endosperm to the embryonal axis. This is completely embryonal axis which has radicle at the bottom and a plumule at the top. Clear, right? So, here you can clearly see epicotyle in both the seeds, hypocotyle also in both the seeds. Hypocotyle later matures to what ma? Radicle, epicotyle matures to plumule. So, here if you can see, embryo consists of what? Cotyledons and embryonal axis. Cotyledons and embryonal axis. Here you see embryonal axis at the base has what? Hypocotyle, which matures to radicle. At the tip, epicotyle, which matures to plumule. The point of attachment between hypocotyle and epicotyle is what? Cotyledons. Clear, right? This is the clear description about parts of the seed. Important. You have to know about the seed coat. What is the outer seed coat composition and inner seed coat composition of a dicot seed, and you have to know about the endosperm, especially starchy endosperm and proteinaceous endosperm of corn, and then you have to know about hypocotyle, epicotyle, radical, plumule, and especially the protective sheets that you see in the monocots. Okay. So, this is the clear description about the structure of seed beta. Now, we will see types of seeds based on the presence or absence of the endosperm. Okay. Types of seeds, based on presence or absence of endosperm. Here we go. Mature seed, if it does not have endosperm, ex-albuminous or non-endospermic seeds, they do not have residual endosperm, as it is completely consumed by the embryo. It is completely consumed by the embryo. For example, pea, groundnut and bee. Next is albuminous or endospermic seeds. Albuminous or endospermic seeds. Most of the dicots are ex-albuminous. Only monocot example that I can, most of the dicots are ex-albuminous. Only dicot which is having endosperm, only dicot with endosperm, dicot endospermic seed. Example I can give you is castor. Okay. Monocots without endosperm, monocot non-endospermic seeds. Example you can take is orchid. Fine till here, any doubt anywhere? Ex-albuminous or non-endospermic seeds, most of the dicots only will be there. Only monocot that I can give you is orchid seeds. Next, most of the monocots are albuminous seeds or endospermic seeds. Examples you can take wheat, maize, barley, and only dicot which is endospermic is castor. Clear till here? Any doubt anywhere? Next, here we go with seed viability. Seed viability is a major concept, and here the examples from your NCERT might be, from your Telugu Academy might be, will be asked. So, here we go. What is seed viability? The period of seed where it can germinate, the period of seed formation, the period from seed formation to seed germination. The ability of a seed to germinate after seed formation, the time period is known as what? Seed viability. So, seed viability, it can be for few months, few days or few years also. It can be for few months. Best example is what? Oxalis. In oxalis, the seed germination can be just for few months. Seeds of many plant species remain viable for several years also. Certain seeds are capable of what? Surviving for hundreds of years. Seeds are capable of surviving for hundreds of years. They are well documented records. We have documented records of this. The oldest known viable seed is what? Oldest known viable seed is lupine. Okay, that is Lupinus arcticus. Lupinus arcticus, which is the viable seed, oldest known viable seed, which is excavated from which Arctic tundra region. Remember the name of this and from which region it is excavated. This seed successfully germinated and flowered after how much period of dormancy? 10,000 years of seed dormancy also it has germinated. during an archaeological excavation at King Herod's palace. This is one more region, King Herod's palace, which is near the Dead Sea. A 2000-year-old viable seed is found. Okay, a 2000-year-old viable seed is found here, which is of date palm, that is Phoenix dactylifera. Okay, how much year old? 2000 year old. And Lupinus arcticus, which is extracted from or excavated from Arctic tundra, after a period of how much dormancy? 10,000 years dormancy also, it has germinated as seed. The fruit of orchids contains thousands of minute seeds. Orchid will contain thousands of minute seeds, like parasitic plants like Orobanche and Striga also will produce numerous tiny seeds. Orobanche and Striga also produce many numerous tiny seeds. So, here till here it is known, right? Ovules post fertilization will turn to what beta? Seed. So, number of ovules will be equal to number of seeds. Here in Orobanche and Striga, how many seeds you can see? Many tiny seeds, plenty of thousands of tiny seeds you can observe in the fruit. Next, fruits. We have three types of fruits based on which from which region fruit formation will occur. Of course, the fruit wall is formed from what? Ovary. Ovary post fertilization turns to fruit wall. Okay. So, if at all any other floral part is also involved in fruit formation, such fruits are called as false fruits. So, here we go with first type is what? True fruit. First type is true fruit, wherein only ovary and ovary is involved in fruit formation over here. Examples you can take mango, kiwi and grapes. False fruit, where other floral parts are also involved in the fruit formation along with the ovary. One example is apple. Well, thalamus is also involved in the fruit formation. Thalamus is involved in the fruit formation. The pulp of apple that we eat is what? Thalamus. The pulp of apple which we eat is what beta? Thalamus. Examples of false fruits include apple, strawberry and cashew. Next is parthenocarpic fruits. The fruits which are formed without fertilization. Okay. Fruits formed without fertilization. Without fertilization. These are what? Parthenocarpic fruits. These are seedless fruits. Okay, they are what, ma? Seedless fruits. Examples you can see banana and seedless grapes. These are three types of fruits beta: true fruits, false fruits and parthenocarpic fruits. And then we see special modes of reproduction: apomixis and polyembryony. From apomixis and polyembryony also we got many questions. Okay, apomixis and polyembryony. Apomixis is a special mode of reproduction. This is a type of sexual reproduction that mimics, this is a type of asexual reproduction that mimics the sexual reproduction. Remember the statement clearly, we can expect the statement based question. Apomixis is a type of asexual reproduction this mimics sexual reproduction. So, first I will explain you the simple phenomena like how the seeds are formed without fertilization, and seeds which are formed with the process of apomixis are addressed as what? Apomictic seeds. Apomictic seeds. These are formed without fertilization. Without fertilization. How they are formed, I will tell you. See. Here we go. First, from the embryo sac, we know in the ovule, in a ovule, let us take an inverted ovule, because most of the angiosperms, 82% of the angiosperms will have inverted ovule, and the cells that are present inside the ovule are addressed as what beta? Nucellus cells. These nucellus cells are what? Diploid. Nucellus cells are of course diploid, right? So, any of the nucellus cell, diploid nucellus cell will directly differentiate into an egg cell, This will develop into embryo directly without fertilization. As such, the diploid nucellus will be differentiated to diploid egg and diploid embryo. This can occur or from megaspore mother cell. Okay. First nucellus will get differentiated, right? Nucellus will be differentiated to megaspore mother cell. Megaspore mother cell which is diploid can be differentiated into a diploid embryo directly, or this can also happen. Any of the nucellus cell or the cells from the integument can also differentiate into embryo, and that can develop into a mature seed. Okay. So, here any of the nucellus cells or megaspore mother cell or integument cells will be differentiated into diploid egg, and this will turn into embryo. Based on what cells from ovule are differentiating, we have two types of apomixis. Okay, we have two types of apomixis. First type is sporophytic budding. In sporophytic budding, either nucellus cells or either the cells from nucellus cells or from the integuments. Cells from the nucellus or integument, they will get differentiated and they will form or they will turn into what? A diploid embryo, and that later matures to diploid seed. Second is what? Recurrent agamospermy. Okay, recurrent agamospermy. Here the diploid megaspore mother cell which is diploid, this gets differentiated into a diploid embryo. Fine, right? Fine till here. Any doubt anywhere? Apomixis is clear, Noma? We have the seeds which are formed as a result of apomixis are known as apomictic seeds, and apomixis is a phenomena where without fertilization seeds are formed. Either the nucellus cell will be differentiated, or integument cells will be differentiated, or megaspore mother cells will be differentiated into egg, diploid egg, which later matures to diploid embryo. Clear till here? Any doubt anywhere? Yeah. Now, here is the concept of polyembryony. Okay. Take some examples of sporophytic budding, beta. Sporophytic budding examples you can take like citrus, mango. Yeah. These are the examples of what? Sporophytic budding. Fine, right? Next you see polyembryony. Polyembryony is first observed or discovered by Leeuwenhoek. In citrus plant. What is polyembryony? Many embryos are present in the nucellar region. Okay, either from the nucellus cells or from the integumentary cells. By the result of sporophytic budding, multiple embryos are seen in the embryo sac, that is known as polyembryony. One nucellar embryo will be there and multiple embryos adjoining to the integuments or nucellus will be formed. That is known as what? Polyembryony. Example is citrus, first discovered by whom? Leeuwenhoek. And now we will get into what? EAPCET questionnaire practice. So far what we have learned today, beta, post fertilization events. First we have looked into endosperm, types of endospermic development and types of seeds based on the persistent endosperm and the absence of endosperm, and types of the endosperms, whether endosperm is absent or the ploidy, that is diploid in Oinothera, and types of the composition of the endosperm. This and all we have learnt under endosperm. Next is what? Embryo development. We have looked into the dicot embryo development, monocot embryo development, which is very important. Have an additional focus over there and learn. After which we have looked into seed formation, fruit formation, types of seeds and types of fruit. Then last two special modes of reproduction, that is apomixis and polyembryony. Now EAPCET questions. Look into it clearly. We will practice few set of questions today and few tomorrow. Look at here. Following has radially expanded cells with fibrous thickenings. This is from the question is from structure of microsporangium. Question is from structure of microsporangium. What are those radially expanded cells with fibrous thickenings? Endothecium, right? So the answer is option B. Next, assertion reason type of question. This is also from microsporangium structure. Structure of microsporangium. Here we go. First, sporopollenin is one of the most resistant organic material called in time. Wrong. This is the most resistant organic material which is found in exine, not in end-time. Assertion is wrong. Exine can withstand high temperatures, strong acids, alkalines and cannot be degraded by the enzymes. True. Okay, so here assertion is false and reason is true, which is option D. Okay. Next you see. Select the correct statements. Okay. Ovary, style and stigma are united in hibiscus. Yes, they are united in hibiscus, ovary, style and stigma. Next, Loranthus shows ategmic ovule. True, Loranthus shows ategmic ovule. Geitonogamy is so. First thing you see, ovary, style and stigma are united. No. Geitonogamy is genetically similar to autogamy. True. It is genetically similar to autogamy, functionally dissimilar. Functionally it is, just a second. Yeah, genetically it is similar to autogamy, but functionally it is cross pollination. Okay, because it is in between two different flowers. Next, entry of pollen tube through micropylar end is called as mesogamy. Wrong. Entry of the pollen tube through micropylar end is known as what, ma? Porogamy, not mesogamy. If it is through integuments, it is mesogamy. So what is the right answer over here? 2 and 3, which is option C. Right. Next, assertion reason type, which is again on the structure of anther. See how important it is. That also it is picked from what? Sporopollenin. Recently asked questions in few years. Sporopollenin is one of the most resistant organic material which is present in the exine of pollen grains. True, sporopollenin can withstand high temperatures, strong acids and alkali and cannot be degraded by any other enzyme. Yes, true. So A and R are true. R is the correct explanation of A, which is option A. Clear, right, beta? Next, last question of today's session. Just look into it and answer. Last question. Yeah. So look into here. Look into it. Find the correct character related to double fertilization of angiosperms. Male gametic nucleus fuses with egg cell. Of course, that is correct. Male gametic nucleus fuses with antipodal cells. Completely wrong statement. Male gamete fuses with synergids. Wrong. Male gamete helps in formation of primary endospermic nucleus. True. By fusing with what nuclei? Polar nuclei or central cell. So here what two statements are correct, beta? A and D. That is option B. Clear till here? Any doubt anywhere, ma? So that's all for today's session. In our next class, we will be dealing with few more set of questions of EAPCET from this chapter. Come prepared, and we will be starting a new chapter, that is cell biology unit, which is very important. Definitely you will be getting 3 to 4 questions from that unit. Okay, so come prepared with this chapter. We will solve EAPCET questions and then we will move on to cell biology. Okay. So, that is all for today's session beta. Thank you.